decades, scientists have successfully treated hormone receptor-positive breast cancer by targeting estrogen signaling. The first generation of therapies focused on the signal itself. Drugs like tamoxifen compete with estrogen for binding to the estrogen receptor, preventing the receptor from activating genes that drive tumour growth. The next generation went one step further. Rather than simply blocking the receptor, drugs like fulvestrant mark the estrogen receptor for destruction. By removing the receptor itself, they eliminate one of the cancer cell's primary drivers of growth. These therapies have transformed breast cancer treatment, but cancer is remarkably adaptable. Over time, many tumours develop ways to maintain estrogen-responsive gene expression despite blocking or even degrading the receptor. Researchers are now asking a different question. What if, instead of targeting the messenger, we control access to the message? To understand this, we have to look inside the nucleus. Nearly 2 meters of DNA are packed into every cell by wrapping it around tiny protein spools called histones. This packaging isn't simply storage. It is one of the cell's most important systems for regulating gene expression. DNA carries a strong negative charge, while histone proteins contain positively charged lysine residues that behave like thousands of tiny molecular magnets holding the DNA tightly against the histone surface. When these molecular magnets are engaged, genes remain difficult to access. KAT6A is one of the proteins that helps regulate those magnets. It does not decide which genes should be expressed. Instead, it acts as an epigenetic co-activator. In estrogen receptor-positive breast cancer, activated estrogen receptors bind specific DNA sequences called estrogen response elements and recruit a team of co-regulatory proteins, including KAT6A, to nearby chromatin. Once recruited, KAT6A acetylates selected lysine residues on histones, neutralizing their positive charge. As those molecular magnets switch off, the DNA loosens locally, making nearby genes more accessible to the transcriptional machinery. In healthy cells, this process is precisely regulated in both space and time. In some breast cancers, however, KAT6A becomes amplified or overexpressed. The tumour becomes increasingly dependent on KAT6A to maintain the open chromatin landscape needed for estrogen-responsive transcription, cell cycle progression, and continued growth. Scientists initially developed drugs that inhibit KAT6A's enzymatic activity. Now, a new strategy has emerged. Rather than simply blocking the enzyme, targeted protein degraders recruit the cell's own protein recycling machinery to remove KAT6A altogether. This not only eliminates its acetyltransferase activity, but also disrupts the larger transcriptional complexes that KAT6A helps organize. The story of KAT6A is not about replacing existing therapies. It is about understanding that cancer can be interrupted at multiple levels. We can block the hormone, we can remove the receptor, or we can remodel the chromatin that determines whether those genes can be read in the first place. That progression reflects an important evolution in precision medicine, from targeting individual molecules to understanding the dynamic biological networks that control cellular behavior.